Hello, Mr. Hunazar Ismail. In section 2, we are going to talk about DNA structure. By the early 1950s, most biologists accept DNA as a hereditary material. However, they still lacked an understanding of DNA structure or how this molecule could replicate, store, and transmit hereditary information and direct cell function. This mystery would soon begin to unravel Cambridge University in England. Now we have DNA double helix. What's the DNA double helix? In the early 1950s, scientists James Watson and Francis Crick begins their work to determine the structure of DNA. So what is the structure of the DNA? In 1953, they made or constructed a model for the structure of DNA, which suggesting that DNA is composed of two chains of nucleotides that wrapped around each other to form a double helix, a shape similar to the winding staircase. Their final model was corrected and remarkable because it explained how DNA could replicate. In 1962, Watson and Crick won the Nobel Prize in Medicine for their work on DNA. This is Watson and Crick. Uh, what's DNA nucleotide? DNA is a nucleic acid made of two long chains, also called strands, of a repeating subunit called nucleotides. Each nucleotide consists of three parts, a five carbon sugar, a phosphate group, and a nitrogenous base. The three parts of DNA nucleotides are illustrated in the figure 85. Let's go to the figure 85. So do you see this is nucleotide? This is a repeating nucleotide. The nucleotide. So we have nitrogenous base. This is nitrogenous base. This is five carbon sugar, and this is phosphate group. This is the nucleotide. The five carbon sugar in DNA nucleotide is called deoxyribose. The phosphate group consists of the phosphorus atoms bounded to four oxygen O atoms. The nitrogenous base contains nitrogen and atom and carbon atoms and is a base. This means they have ability to accepting hydrogen. This is what they are base. Bonds, what's the bonds that hold DNA together? The DNA double helix is similar to a spiral staircase. The alternating sugar and phosphate molecule form a site of handrails of staircase. Nucleotides along each strand are connected like covalent between the sugar of one mononucleotide and the phosphate of the other phosphate group of the mixed nucleotide. Each full term of the DNA helix has 10 nucleotide pairs. The nitrogenous base, called bases in, for short, face toward the center of DNA molecule. The base on one strand of DNA face and form a bond called hydrogen bonds, with the base on the other strands of nitrogenous base are bonded in the pair between two strands by two or three hydrogen bonds. The base pairs forms steps with the staircase. The base pairs are uniform width because in which pair in each pair, one base has a two-ring structure and the other has a single ring structure. And the dashes lines indicate the location of the hydrogen bonds. Hydrogen bonds between the bases help hold the two chains of the DNA helix together. Now look at this figure. This is the hydrogen bond. Now between C and G, we have uh, three hydrogen bonds. But between A and T, we have two hydrogen bonds. So this is nucleotide, which is this is nitrogen base, this is five carbon sugar, and this is phosphate. This phosphate bind to this sugar in this nucleotide, and this phosphate bind to this sugar in this nucleotide, and so on. So the phosphate and sugar making this line, and the nitrogenous base is in the middle. So what's the nitrogenous bases? The sugar and phosphate group are identical in all DNA nucleotides. However, the nitrogenous base may be any one of four different kinds, maybe thiamine, cytosine, adenine, or guanine. Nitrogen bases and their chemical structure called rings. The nitrogen bases are often represented by the first letters for their name. T for thiamine, C for cytosine, E for adenine, and G for guanine. Nitrogenous bases that have a double rings of a carbon, a nitrogenous atom, such as adenine and guanine. So the adenine and guanine are purines, they are double bonds. Nitrogen bases that have single rings of a carbon and nitrogen atoms, such as cytosine and thymine, are called pyrimides. They have single rings. So this is double rings, and this is double ring purines and double single ring pyrimides. Always one purine in one strand binding to the pyrimidine to the other strand. So what's the complementary bases? Complementary base in 1949, American biochemist Erwin Chargaff observed that the percentage of adenine equal the percentage of the thymine, and the percentage of cytosine equal the percentage of the guanine in the DNA of a variety of organisms. This observation was key to understanding the structure of DNA because it meant base by base pairing rules in DNA. Cytosine on one strand pairs with the guanine on the opposite strand, and the adenine pairs with the thymine. These pairs of a base are called complementary base pairs. Notice that each complementary base pair contains one double ring purines and one single ring pyrimides. Because of base pairing rules, the order of the nitrogen base on the nucleotides in one chain of DNA molecule is complementary to the order of the base on the opposite chain. For example, if a DNA chain has the sequence ATTC, so then the other chains must have the complementary sequence TAAG. The order of nitrogen bases on a chain of DNA is called base sequence. Complementary base pairing is important in DNA structure and function for two reasons. First, the hydrogen bonds between the base pair help hold the two strands of DNA molecule together. Second, the complementary nature of DNA helps explain how DNA replicates before cell division. One strand of DNA molecule can serve as a template for making a new complementary strand. So DNA model, this is the first DNA model. Then it's going to be simplified, making this type. Finally, they are making this model, only writing the nitrogenous bases. DNA double DNA models. What's the DNA models? The structure of DNA is often simplified when it is drawn or modeled. For example, the DNA double helix is often illustrated as a straight letter. The sugar phosphate handles are drawn as a straight line so that these pair steps between the DNA strands are easier to see. You see one line because the sugar atoms are the same. So just we will write down one line. This line representing sugar and the steps is what is the nitrogenous bases. Notice that simplifying DNA structure highlight the complementary base pair in each of the DNA nucleotides. In some cases, the structure of DNA is simplified even more by just writing the first letter of each of the nitrogenous base in the DNA nucleotide. For example, the DNA, it will be the model will be like this. Okay, for easy. So nowadays the model is right down. So this E is representing not only the nitrogenous base, but the whole nucleotide. Thank you.